Good afternoon, everyone. I am Hannah Erica F. Gumayen from BS Fast 3A. Today, I am going to discuss the different parasitic microsporidian on farmed crustaceans, Penaeid shrimps, and crabs. Shrimps are one of the most popular aquaculture species in the Philippines. However, disease outbreaks over the last 2 decades have led to dramatic declines in the shrimp output and challenges for the Philippines claiming its place as a global shrimp leader. One of the known diseases is caused by a protozoan called Microsporidia. Microsporidia are highly specialized obligate intracellular eukaryotes that are closely related to fungi. They have a relatively simple development consisting of two general stages: merogeny and sporogeny. Meronts multiply inside the infected host cell, eventually forming sporons and then spores, which are ultimately released from the host and transmit the infection. There are almost 200 genera and over 1,400 described Microsporidian species, though the actual diversity is estimated to be much higher. Diverse pathological effects of Microsporidia have been described in animals, especially in livestock such as crabs, lobsters, and shrimps. Their impact on crustaceans' health is well acknowledged, either producing direct mortalities, diminishing catch value, reducing growth rates, increasing the chances of predation by other animals, or impairing the reproductive or the cardiorespiratory capacities. This study aims to define parasitic microsporidians, identify the different parasitic microsporidians and farmed crustaceans, and to discuss diagnosis and effect of parasitic microsporidians and farmed crustaceans. Common parasitic microsporidians on crustaceans. Common name is microsporidiosis, also known as white ovaries or microsporidian infection. Causative agent is Microsporidia. Species affected are Penaeus monodon, Penaeus merjuensis, and Penaeus indicus. The stages affected are usually juveniles and adults. Gross signs shows that affected tissues and organs turns opaque white. As you can see in the image, the white tissue or organ of shrimp turns opaque white, while its effect on host is that spores and other stages of the parasite replace the affected tissues. We can see it in the second image. The microsporidian infection of the abdominal muscles of Penaeus indicus and ovary of Penaeus monodon. The tissue is replaced with the parasite. Infection may result in sterility of spawners with white ovaries. Lastly, infection rate is relatively low, but the parasite is highly pathogenic. Second parasitic microsporidian that I will be discussing is Enterocytozoan hepatopenae, or also known as EHP. The species known to be susceptible to EHP are black tiger prawns, Penaeus monodon, Pacific white shrimps, Penaeus litopenaeus vanamae, and Pacific blue shrimp, Penaeus litopenaeus stylirostris. What is EHP? Enterocytozoan hepatopenae, also known as EHP, is a microsporidian parasite that was first characterized and named from the giant or black tiger shrimp Penaeus monodon from Thailand in 2009. It was discovered in a slow-growing shrimp but was not statistically associated with slow growth at that time. EHP is confined to the shrimp hepatopancreas or HP and morphologically resembles an unnamed microsporidian previously reported in the HP or hepatopancreas of Penaeid japonicus from Australia in 2009. Epidemiology: hepatopancreatic microsporidiosis is caused by infection with enterocytozoan hepatopenae or EHP. It is a microsporidian parasite classified within the family of Enterocytozoonidae and was first discovered in Penaeid in Thailand in year 2004. The spore ultrastructure of the parasite also resembled another uncharacterized hepatopancreatic microsporidium previously reported from Penaeid in Malaysia in 1989 and from the PL of disease Penaeid japonicus examined in Australia in 2001. The early studies indicated potential for infection with now known EHP-like microsporidians in several Penaeid shrimp species, leading to the proposal that EHP-like microsporidians were endemic within the Australian Asian Pacific region prior to the expansion of shrimp aquaculture in the 1990s. Therefore, we can say that EHP is already common in the year 1990s or in late 1990s, although it is only discovered in Penaeid in year 2004 in Thailand. Next is the life cycle. The general life cycle of a microsporidian can be divided in three distinct life phases: an infective spore phase, a proliferative phase, and a spore-forming phase. Microsporidia are mainly transmitted via oral ingestion of the spores, although vertical transmission has been demonstrated in some species. The spore is a free-living stage that can survive outside the host for several years. It has a unique extrusion apparatus termed the polar tubule that, when given adequate stimuli, averts and pierces a suitable host cell to deposit the sporoplasm. Once inside of the host cell, the proliferative phase and eventually the formation of mature spores occur. Here is an example of a life cycle and its transmission routes from Science Direct. As I have said a while ago, microsporidia are transmitted via oral ingestion or through sexual intercourse between contaminated species or infected species and non-contaminated species. In oral ingestion, because an infected species is living in a tank, same with an uncontaminated shrimps, it is possible that the infected shrimps can contaminate the water of the tanks and can infect other shrimps inside the tanks through EHP spores in feces. While the life cycle, we can see that merogeny multiply inside the infected host cell. Then, sporons eventually forms, and then spores mature. Eventually, the formed sporons become mature spores. Diagnosis of microsporidians. Microsporidians is mainly based on the use of molecular diagnosis methods and light microscopy to detect the spores. Microscopic visualization of microsporidians in tissues sections can be difficult due to the small size of the spores. A 1 to 20 micrometer. As a result, special staining methods such as modified trichrome stain, Gram-Wharton, starry silver, or calcafluor white may be required. More sensitive methods like immunohistochemistry or in situ or situ hybridization are available for certain microsporidian species. Here are different images wherein hepatopancreatic tissue are diagnosed through hematoxylin stain, trichome stain, H&E stain, toluidine blue stain. In image A, we can see that H&E stained smear of hepatopancreatic tissue showing numerous microsporidian spores. In image B, fresh preparation of microsporidian spores from a Percol gradient can be seen. While in image C and D, hepatopancreatic tissue section showing acidophilic granular inclusions in the cytoplasm of tubule epithelial cells. Lastly, in the image E, is a semi-thin section of hepatopancreatic tissue showing early and late plasmodia and mature spores in the cytoplasm of tubule epithelial cells. Some spores show unstained spots that represent their concave surfaces at one end. Methods of detection parasitic microsporidian. Detection of parasitic microsporidians on farm crustaceans. The endoparasitic protozoans that causes the affected tissues or organs turn into opaque white can be diagnosed only through microscopic examination of the infected tissue. Thus, we're using a staining method for detection of EHP spores with calcafluor white can be seen in the image below from Science Direct. A Penaeid vannamei, susceptible for having an infection of EHP, can be diagnosed, or we can detect EHP in it through getting a feces from its organ or tissue, affected tissue. We can use CFW stain to detect whether EHP spores are present. And as we can see in the image, the blue-white fluorescent EHP spores can be seen. Prevention and control of EHP on farmed crustacean. PL management. First, you need to confirm with PCR test that PL is not infected. Ensure no EHP contamination of live bait during selection stage. Instrument disinfection means before using an equipment, you need to disinfect it. Environment and water quality management involves increased sewage discharge capacity to avoid deterioration of the water quality in the pond. Add probiotics to the water to avoid proliferation of pathogens or bacteria. Lastly, avoid inappropriate use of medicine lest it cause hepatopancreatic damage. Disease management also involves usage of uncontaminated feed to enhance immunity. Dead shrimps should be removed as possible to avoid cross-infection. Use high-quality SPF shrimp PL based on PCR testing protocols. Why is EHP important? Although EHP does not appear to cause mortality, information from shrimp farmers indicates that it is associated with severe growth and retardation in P. vannamei. Scientists now have information indicating that EHP outbreaks are occurring widely in China, Indonesia, Malaysia, Vietnam, and Thailand. Very recently, they also have received samples of PCR positive for EHP from slow-growing shrimp in India. Thus, EHP is an emerging problem that is under urgent need of control. So the importance of EHP is that scientists or farmers wouldn't detect whether their farmed crustaceans or farmed shrimps are infected with EHP. Lastly, Hepatospora eriochare. I will be discussing this parasitic microsporidia that infect invasive Chinese mitten crabs known as E. sinensis. As we can see, this is an image from ResearchGate. It is a image of H. eriochare in an electron microscopy. The type of species in the image is H. eriochare. It has spores ellipsoid approximately 1.8 to 0.9 1 micrometer in size in tissues fixed for transmission electron microscopy. It possesses 7 to 8 polar filament coils in a single rank. All life stages are eukaryotic and contained within an interfacial membrane separating life stages from the host cell cytoplasm. Presence of a microsporidian parasite with descriptive features of the above in cytoplasm of hepatopancreas of the host. Diagnosis of morphological features by histology and transmission electron microscopy. The type of host is E. sinensis, or also known as Chinese mitten crab. Type of locality is China. Site of infection is cytoplasm of hepatopancreatic epithelial cells, distinct from the nuclear location of the crab-infecting genus Enterospora. Etymology, the generic name refers to the location of the parasite in the hepatopancreas of the host. The scientific name refers to its infection of the host, E. sinensis. Type material is that the whereabouts of syn-type specimens from China are unknown. The image is a proposed life cycle of Hepatospora based on histopathological examination from ResearchGate. As we can see, the first stage is merogeny, then the formation of sporons and then spores. How to prevent and control Microsporidian on farmed crustaceans, specifically on crabs. Avoid feeding stocks with contaminated feeds. disinfect the culture system using chlorine or iodine solution, ensure good water exchange, and isolate and destroy disease stocks. Physical, environmental, and chemical methods may also be used to prevent and control parasitic diseases. The physical methods may involve the removal of the sources of the parasites or subjecting the parasite to adverse conditions like increased or low temperature. Scrub the tank thoroughly with a brush and filter the rearing water with a fine net. Environmental methods may also include proper hatchery or pond design, good water quality, employing sanitary practices, and avoiding stocks from being stressed. Installation or installation of disinfection rugs and trays at the entrances of hatchery facilities. Chemical methods may also include the disinfection of rearing water, feeds, and other materials used for culture, and chemotherapy, which employs drugs or chemicals for treating parasitic infestation. When dipping infected crabs in a chemical bath for a short period, add the chemical solution to the holding tank with the species. Let the species remain in the solution depending on the recommended soaking time for the chemical use. After treatment, either replace the tank with clean rearing water or transfer crabs into parasite-free tanks. Definition of terms. Clinical signs. It means changes in the normal healthy state, bodily function, or behavior of species observed. Diagnosis. The identification of the nature of an illness or other problem by examination of the symptoms. Epidemiology means it deals with the incidence, distribution, and possible control of diseases and other factors relating to health. Enterocytozoan hepatopenae is a microsporidian parasite that was first characterized and named from the giant or black tiger shrimp Penaeid monodon from Thailand in 2009. Hepatospora eryochere is an emerging microsporidian pathogen for the Chinese mitten crab E. sinensis. Microsporidia is an order of spore-forming protozoans from class Microsporidia that are parasites of various invertebrate and vertebrate animals that typically invade and destroy host cells, and that includes some enterocytozoan and hepatospora. Pathogenesis means it is the manner of development of a disease. In conclusion, based on the results and discussion, microsporidian are highly specialized obligate intracellular, intracytoplasmic, and or intranuclear eukaryotes that are closely related to the fungi. They have relatively simple development consisting of two general stages, merogeny and sporogeny. In line with this, a significant example in microsporidians affecting farm crustaceans, specifically in Penaeid shrimps and crabs, is enterocytozoan hepatopenae and Hepatospora eryochere, an emerging parasite and critical threat for farm Penaeid shrimp and crabs in Asia that causes severely retarded growth. Lastly, microsporidians are mainly based on the use of molecular diagnosis methods and light microscopy to detect the spores. The effects of these parasites or parasitic microsporidians to the host is that the spores and other stages of the parasite replace the affected tissues. It also affects the growth of the species or host. Infection may result also in sterility of spawners with white ovaries, and lastly, infection rate is relatively low, but the parasite is highly pathogenic. Here are the references of my presentation. And before ending this presentation, I also want to play a video about the detection of EHP using wet mount microscope. microscopy, rather.
Poor growth. Most of the shrimp farmers do their first sampling between 30 to 40 days of culture. During this time, if you find poor growth, very high size variation, and certain percentage of shrimps with loose shell, you can consider this as a suspected case of EHP, enterocytosome hepatopenia, a fungal disease. There are two methods to confirm it. The first one is PCR. It is very sensitive. But it is costly and time consuming. The second method is wet mount microscopy. It is very easy to do it, and you can do the test within 5 minutes. Today in this video, I am going to demonstrate EHP detection with wet mount microscopy. Select the small shrimps from the suspected pond. Remove the carapace and expose the hepatopancreas. Take a small piece of hepatopancreas with the help of forceps. A small piece is enough. Place it on a glass slide. Add a drop of water. Place cover slip over it. Gently press it. Now your slide is ready. Observe the slide under either 40x or 100x oil immersion. But I am going to observe only under 40x, and this is the video taken at 40x magnification. And now you can see EHP spores. EHP is an intracellular pathogen, especially it is intracytoplasmic. You should be able to identify the cell, you should be able to identify the nucleus, and the EHP will be between in the cytoplasm. It won't be there in the nucleus. So there are several spores here, and this is a very severe case. You can see all the EHP spores spread. This is a very, very severe case. If you find shrimps like this, it is better to harvest. If you do real-time PCR, it may be positive at 14 CT. A very high number of spores. All the spores are uniform in shape. It's easy to identify. And you should be able to differentiate EHP infected cells and normal cells also. It's very easy.
So that's the end of my report. Thank you for watching and thank you for listening. God bless.